Hello, welcome. This is Mrs. Rowe, and welcome to our Biology from Home series. And today I am going to be walking you through how to build your cell membrane models. So you should have these packets from before we left school. However, I did upload a scan of these. If you have accessibility to a printer, you can also print this out. So this is the build a membrane model, so that today's our off-screen day, hopefully, that you can do something that doesn't involve a computer so much. And as you can look, it's got some instructions of what your model should look like when you're done. So don't forget to refer back to this if you get stuck on what you want it to look like. A reminder, if you've gone through some of the videos, that we're looking at the cell model mosaic theory, and that the idea is that it's a double lipid membrane with a mosaic of proteins and other molecules that are embedded throughout, and that this is a flexible membrane that creates that barrier between the internal and external environment for our cells. And as a single layer membrane, it also creates the membrane-bound organelles that we have in our eukaryotic cells. So what I have done, just to do a quick walkthrough with you, is I've pre-colored and cut things out, so you don't have to sit and watch me do that. So we've used models before, if you remember when we did our protein models and we built proteins. And if you remember, we used the color yellow like butter to represent nonpolar. Quick review, nonpolar means it's hydrophobic. It does not like water. It has no charges to it, whereas polar, like water, has both positive and negative charges on it, making it hydrophilic. Polar likes water, so I did blue for polar, showing that that's the side that's attracted to water. And I did yellow for nonpolar, showing that that is the part that is going to move away from water. So it's going to, these phospholipids, which is just one ball with two legs coming off of it. If we could do one phospholipid, just a reminder, I'll do it up here on the board. One phospholipid. We built some of these, if you remember, with our marshmallows. It's got that phosphate head that's polar and the non-polar tails. Well, those tails are automatically going to kind of turn and rotate away from the water that's inside the cell in the cytoplasm and in the external environment. So when you cut it out, make sure that you follow those heavy dark lines. Notice I did not cut out the end pieces. That's the number one mistake. If you see a dotted line, that's a fold line. Do not cut the fold lines, only cut those heavy dark lines. So it should look like this once you have finished cutting it. So first we're going to put the main part together. Then you have your proteins, and I've used pink for the polar parts of the proteins. You don't have to color everything. I just wanted to put a little bit of color in there. And again, I've covered, don't cut any of the dotted lines. Those are your fold lines. Only cut on the outside of the heavy dark lines. So to begin with, we're going to fold on those dotted lines so that we can start to create, this is going to basically create a box. So I'm going to be doing it quickly because of the sake of the video. You do not have to have it perfect. Get the big idea here. Again, following those dotted lines to make this box. And then I've got another one there. Normally when we make these in class, everybody makes one, and then we connect them all together and we end up with a giant cell membrane. It's actually pretty cool. You guys will just be doing the single piece so that you get to see and think about the membrane in a 3D manner versus that 2D diagrams that we usually get. So you can see already we're starting to get, if I tape this together, I'm starting to get the box shape. Now I got to do the ends. So I'm going to fold those ends. And get those folded on those dotted lines. The hardest part working with this is these little tabs that they use for taping and joining. Trying to keep those ready to go. And of course you're going to need some glue or some tape. And I know it's hard. You might not even have scissors or things to color with. You can use crayons, you can use markers, you can use, I like highlighters because then I can still see the structures through them. Do what you can. If you can't color it, you can't color it. If you don't have tape or glue, you can make your own glue with a little bit of flour and water. When I was a kid, that's how we pasted things. We made paste by just mixing flour and water. Just enough water that you're making like a paste You don't want it too thin, and then you just kind of brush that paste on and let it dry, and it will work. And I made a lot of projects as a kid using flour and water. Okay, so the first thing we want to do is we want to tape these things together to form that box shape. So this one here, you're just folding it till you get that box shape that we're looking for. And I'm just going to put some tape on there. Tape is a little bit quicker. Making sure that it kind of matches up with, you can see how that's going to tuck in when I do this. I don't want to tuck in too far to make this box. Okay. I don't know if I can get one. I'm not going to worry about it. Tucking it in there. I don't have to tuck it in. I could tape it that way. So it really doesn't matter. The idea is that, oops, I probably should have used this as my guideline. Get that all lined up. Get that taped. It's not going to be perfect, if that's okay. I can use this to kind of pull that out a little. Okay, so I'm just doing it quick, but you get the idea. There is my 3D, the top of my cell membrane, all the phospholipids there that attract water. And the reason why is this semi-permeable, because small nonpolar molecules such as oxygen and CO2 are going to be able to diffuse directly through this because they're so small, and they're able to pass this nonpolar center part of the membrane. Things that are large, like sugar and proteins, will not be able to pass. Even water is not going to be able to pass because, again, this hydrophobic center. So how are we going to get things in and out of this membrane? Well, we have proteins. I'm going to start with a channel protein, and proteins allow, this one will allow small polar molecules to pass. And again, we're going to fold this on the dotted line. So I'm just going to do one of these and then to show you how they go on. And don't forget, you can then go back to this as a diagram to help you put the rest of it together. Okay, I think you're getting the hang of it here, but I want to show you one of the proteins and how they kind of fold around. So I'm just making all of these little folds. And then I'm going to put this on. Where they actually attach is up to you. It's a mosaic. They can go wherever you want to go. They are not in permanent position. They are actually able to move around across this membrane. Okay. Probably the most exciting video you've watched in a long time. Mrs. Rowe folding paper. Yes. Okay, almost there. This one's got a lot of folds to it. You are going to find one of these does not have a lot of folds to it, other than this, which comes across, but you're going to see that these typically match the width there. So most of these are going to, even something like this, they're going to form these little circles as they come around, and this is going to come across on top. So when you tape this guy together, so some of these seem to be missing little folded ends so that you can bring them around like this. Bring this guy around, and notice then that this can kind of come and cover the top of it. So as I bring the top down, you get the idea. I could play with it. I'm trying to do it fast. You can try to. Okay, now I have a protein stuck on my thumb. There we go. Okay, so it's not perfect. I'm just doing it fast, but you get the idea. It's making like a little cylinder. And then this one's got a little protein hanging off of it. So then what I would try to do, my tape's getting in the way, is I would just find a place to attach it and tape it on. Okay, so this is one of my little tethered proteins. And this little looks like a phospholipid. Maybe it's probably a cholesterol, maybe. This little lipid shape, and there's one of my proteins attached on top. So you get the idea how we're going to start to attach these. So the same thing. These guys are going to fold around, and as they do, they're going to create these circular shapes to try to make them 3D. So I'll take this guy over here. This gets folded down, and then you've got all these little tabs that fold in to help hold it. Again, you could have the tabs on the inside, outside, it doesn't really matter. We're not looking for perfection, we're just trying to get the big idea of what this looks like. This is where sometimes maybe glue would be a little bit easier, I don't know. So you could glue all of these different tabs in place. This one over here, it's not quite bending perfectly, but that's okay. So you can see there's the top of my protein. I'll do the bottom part over here. I bet you're all thinking, yes, I can't wait to do this. Okay, so there's my protein for my channel protein that's going to allow small molecules to come through. I can put this on the end, I can put this wherever I want. Notice how this little side piece here is made to fit the width of your box. If everything's been folded correctly, hope it's supposed to fit. Maybe I didn't quite fold it quite right, but normally that would fit there. You can see that I'm struggling to get these in, but the idea is you would do that, and then I would take a piece of tape and put that on. So you probably want to take a little more time and care than I'm doing. I'm kind of rushing it, which is making it a little bit harder for me to get these guys on. But you get the idea. So I've got this guy attached. Ideally, I should be able to attach it here. And this bottom piece should just, I don't know why it's not quite fitting, but that bottom piece should be able to come across. Creating our little channel protein. So there's our channel. That would be channeling. Ideally, there'd be two of these. This is 1/2. That channel would be through the middle. So what I would do is take someone else's model and put them side by side so that we create a full round protein with the channel through the middle. This is like a protein cut in half. But maybe I cut mine too quickly. Maybe I didn't fold it right, but normally these would fit. If I had time, I could trim it and make it fit a little bit nicer, but that's the idea. When you've got your whole protein assembled, take a picture and then do what you will with it. You've got a nice little 3D cell membrane, and if you want to save it and bring it to school, we'll put them all together once we get back to school. I hope that helps. Hope you enjoy building your 3D membrane. Hopefully it's not too frustrating for you, and until next time, have a great day.